Welcome to the LD50 serial dilution toxicology lab using Lactuca sativa, which is red leaf lettuce. The purpose is to determine the concentration of sodium chloride that will kill 50% of a given population of Lactuca sativa seeds. In short, to find the NaCl LD50 for Lactuca sativa seeds. There are many ways to evaluate health threats from toxic substances in the environment. Toxins may affect cells, tissues, organs, organ systems, whole organisms, populations, and ecological communities. Toxins may be either synthetic or naturally occurring. In the science of toxicology, the study of toxic substances, bioassay techniques are often used to evaluate the direct lethal effects of substances on organisms. A bioassay is a test that uses living organisms as an indicator. A common measure of acute or lethal toxicity is called the LD50, lethal dose 50. This is the experimentally determined concentration of the substance that will kill 50% of the individuals exposed to it. The reason that some, but not all, of the exposed individuals are killed is twofold. First, there's genetic variation among individuals in a population. Some individuals are more susceptible and some are more resistant to the lethal effects of any substance. Second, exposure to the toxin is uneven. The toxin is likely not to be evenly distributed in the water or environment of the individuals. One of the challenges of a good experiment is making sure that all the experimental individuals are evenly exposed to the toxin. Also, it is extremely important to remember that different species will vary in their response to a particular toxin. What may be extremely poisonous to one organism may have no harmful effects on another. That is why test studies using lab animals cannot be extrapolated to humans with 100% certainty. Generally speaking, the more closely related two animals are, the more the results can be extrapolated from one to another with confidence. Universities and government agencies use vertebrate animals such as rabbits, rats, and chimpanzees for drug and toxicology testing. We'll be using the seeds of red lettuce from my garden. Lactuca sativa is good for this experiment because it is easy to see and count. Lettuce seeds are also frequently used to test water and other environmental samples because of their sensitivity to environmental contaminants, such as heavy metals and some pesticides and solvents. For this lab, you will need some salt. You'll need some water. You'll need tablespoon and teaspoon, and you'll need a pipette. You'll need some baggies. And you'll need the lettuce seeds that I gave you in your packet and some paper towel. Don't forget that. So you're going to need 8 baggies or 8 containers. These are just to keep the lettuce seeds moist so that they're not going to dry out. I'm going to use baggies. So make sure that you label them with the follows: control, 1 part per million, 10 part per million, 100 part per million, 1,000, 10,000, and 100,000 part per million. Here are a couple of my baggies that are going to be used. You also need some paper towel. You should double or triple up the paper towel. The paper towel needs to fit inside of your baggies. So this one I'm going to have to cut a little bit in order for it to work. We're also going to need to make some different solutions. We're going to need to make solutions for each of these little baggies. If you remember, we learned that 100,000 parts per million is the same as a 1-to-10 ratio. So in order to make this first most concentrated solution, what you're going to do is you're going to have a container, and in that container, you are going to put 10 tablespoons of water. And to that, you're going to add 2.5 teaspoons of salt. 10 tablespoons of water and 2.5 teaspoons of salt. I had a 1/2 teaspoon measure, so I needed to add 5 of them to get 2.5 teaspoons of salt. I'm going to stir that up until it's all the way dissolved. That is going to be my 100,000 parts per million solution. From there, I'm going to dilute it just like the serial dilution lab that we did. Only instead of drops, we're going to use tablespoons. So into my next container, I'm going to add 1 tablespoon of this and 9 tablespoons of water. This is going to be my 10,000 part per million solution. I'm going to continue to do that until I get all the way diluted to 1 part per million. So there's 9 tablespoons of water and 1 tablespoon of my super salty water. So next you need to put it all together. This is my control. I'm going to get this paper towel a little bit wet with just plain water. Onto that, I'm going to add 10 lettuce seeds. Kind of make them like stick down. Make sure that they're going to stick. They don't fall over everywhere. Now, the lettuce seeds are mixed. They are from my garden. So they're mixed with some garbage, like just some plant material. So this is what those seeds look like. Okay, they each are an individual seed. I'm going to put these into my baggie. I'm going to add a little bit more water because I want to make sure that they've got plenty of water, that they're not going to dry out as I let them grow. If you don't have a bag, you just have like a Tupperware container or something like that, that's fine. Cover it with something so that it gets light inside. These guys are going to want some light. Okay. But seal it up so they don't dry out. So I have my control done. Now I'm going to do the same thing for each of the 8 different ones. But this time, instead of using water, I'm going to use my really salty solution, my 100,000 parts per million. I'm going to get it a little wet so that the seeds stick and don't fall all over the place. And then I'm going to add my 10 seeds. I'm going to put this into the baggie. I'm going to add just a little bit more water. And I should say this isn't pure water, this is my solution that I made. I'm going to make sure that this is sealed, and I'm going to lay it somewhere where it's going to get some light. It'd be a good idea to clean my pipette before I put a new solution on. Here's my 10,000 part per million solution. Again, I'm going to get it a little wet so that the seeds stick. And then I'm going to put the seeds on it and put it into the baggie. And add just a little bit more, making sure that it's plenty wet. So here are the 3 I've just made. You will make 8 total. You're going to let them sit somewhere where they can get light for 4 days. So after the seeds have germinated, after about 4 days, you're going to count the number of seeds that have germinated and measure the length of each radicle, which is the little embryonic root. You're going to carefully remove the whole seed from the paper towel in one piece. Be careful not to pull too hard, and measure the length to the nearest millimeter of this radical. You're going to calculate the mean or the average radical length for each solution. Do not include zeros for the seeds that did not germinate. Here are the 2 charts. Here's the first chart. You're going to just fill in one of these columns of how many died. How many died in the control, in the 1 part per million, in the 10 part per million, what percent died. Then you're going to fill in one of these tables with the average radical length. If none of these germinated, then you're just going to have a 0 there. There's not going to be any data. But if all 10 of these germinated, you're going to average the length of those radical roots. So it's been 4 days. We're looking at the 100,000 parts per million seeds. And you see that none of them have germinated. There's nothing growing out of any of them. So we can assume that all of them died. So for the 100,000 parts per million, the percent mortality would be 100%. Here's the 10,000 parts per million. And for mine, the same is true. They all died, 100% mortality. Here's my control. All of my control germinated. They all started growing. So I'm going to write 0% mortality. You are going to complete yours. I'm probably sure that these are going to be the same, but go ahead and report your data. But make sure you get all of these. These are the ones that are a little bit questionable. Then for each one that germinated, you're going to carefully remove from the paper towel. They might have grown kind of into, so they might stick a little bit, but you're going to remove from the paper towel and you're going to measure how long that radical length is in millimeters. Okay. You're going to do that for all the ones that germinated, and then you're going to average those together. Here was my water. They all germinated, so I had 10 numbers, and I got an average of 9.8 millimeters for the radical length. You're going to do the same for all the others that germinated in 1 part per million, and then again in 10 part per million. If they didn't germinate, they're not zeros, they're just not a number. So after you have your data, you're going to have it written out in a table similar to this. In the description of this video, I will put a link of where to report your data. I'll also put 2 links on where to get class data, because that will be useful once you go to graphing. So let's talk about graphing. There's 2 different kinds of graphs you're going to need to make. The first graph is going to be using logarithmic paper. This is not logarithmic paper. This first graph is going to be using mortality data. What percent died? This mortality data you're going to get from the link below, the overall class mortality data. And you're going to use this big logarithmic paper graph in order to graph this. On the bottom, you're going to have percent mortality. It's already marked for you. 0%, 20%, 40%, 60, 80, 100. 50% mortality is a little bit darker of a line because we're going to need that when you're all done graphing. On the y-axis, you're going to start with 1. You're not going to graph the control for this one. You're going to start with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This will be 10 right there. After the 10, this is going to be 10, 20, 30, 40, 50, 60, 70, 80, 90, 100. 100, 200, 300, 4, 5, 6, 7, 8, 900, and 1,000. One thousand, 2,000, 3, 4, 5, 6, 7, 8, 9,000, and 10,000. 10, 20, 30, 40, 50, 60, 70, 80, 90,000, and 100,000. I think you have to draw the 100 on there. So, what percent of 100,000 parts per million died? I'm thinking it's probably going to be close to 100%. What percent of the 10,000 parts per million? You're going to graph that dot. What percent of the 1,000? What percent of the 100? Of the 10 and of the 1 part per million? Once you have all those points, you're going to try and draw a line. From last year's data, it seemed that there was a threshold where it didn't seem to matter with the increase of concentration. There was just the same amount of mortality. But then it seemed to change, and I drew a line of best fit for all the points that I had. Now, after you guys get this year's data, these numbers are going to change a little bit. So I'm just showing you an example right now. Once you've graphed this line of best fit, I want you to zoom in on the place where it crosses the darker line. The darker line is the 50%. We're trying to figure out what is the LD50 for salt for these seeds. What is the concentration that 50% of them die? So where my line crosses that darker line, you're going to follow this all the way over to the other side, and you're going to say that LD50 for this particular period, I got 810 parts per million. Okay. Yours is going to be a little bit different. I did this 3 times, 3 periods. I got 3 different numbers. Yours is going to be a little bit different. So I want you to give me the LD50 for the numbers that you graphed. I would suggest you graph the class average numbers, which are going to be in a link in the description below. So this is how you're going to graph the mortality data. Next, you're going to graph the radical length data. This is going to be on a normal piece of graph paper, a piece of graph paper like this. There's also going to be a link down below for class average data. I would suggest to use that link. Now, how are you going to decide how to set up this graph on normal graph paper? Here's my hint. There's this thing called mix dry. Mix is manipulated, independent, and x-axis. All those are the same thing. And dry is dependent, responding, and y-axis. What we're looking at here is how does concentration affect radical length? How does concentration affect radical length? The thing that you're changing is concentration. The thing that you are measuring is the radical length. So the concentration is going to be on your x-axis. The radical length is going to be on your y-axis. So you're going to make a graph with concentration on the x-axis and radical length on the y-axis. Now, why do we have 2 different graphs? Well, this is going to be a linear relationship. So we're going to use normal graph paper. LD50, however, it's a logarithmic relationship, which means that you can't estimate unless you try and flatten out that line. If we were to graph this same data on graph paper like this, we would get a logarithmic curve and we wouldn't be able to estimate LD50. We have to straighten out the line by using weird graph paper in order to estimate the LD50. This is the reason why we have 2 different kinds of graph paper for this lab. Then you're going to answer these questions for the conclusion. Good luck. Have fun finding out what is the LD50 of salt for these red lettuce seeds.